As usual, I try to bring in some degree of cytology for you. So essentially, it's explaining the concept of what the topic is and then trying to put in some perspective of histology and cytology for the sake of imaging. And so today we will look at this topic, and I will have a little bit of interspersed some videos for you regarding some techniques which is within this topic. So invasion and metastasis. Yesterday, so far, we've studied, we learned, or you learned, that the cells, they learn how to, they pick up all the mutations and then they start to multiply. And then they do grow into some kind of a tumor, but they don't really invade and they haven't spread. So once they have grown big, what is it that they do to spread? Sometimes they don't. They don't invade the other tissues and they do not spread. And so if they don't spread, then everything is fine and dandy. There has to be a small surgery and they can be removed. And that's a good news. But if they start to invade, then these cells are smart. What do they do to invade and what do they do to spread? So really, our first couple of really what we want to talk about is just spread. You know, what is it that is, what does it mean to be just spreading? So in terms of spread, the words that you should know is that there's a primary cancer. That is where the cancer starts. Okay, that's the beginning, where it starts and where the cancer has grown. Okay, and if only they spread to other parts of the body and they start to form a tumor there, only then will it be called as the secondary cancer. So the secondary cancer is only when that tumor has grown. Sometimes they invade and tumor doesn't form. You know, it takes many, many years, or they invade, the tumor cells don't sit. So the spread of cancer really depends on that cell to be able to go and spread and be successful. So they can, so therefore the word is that they can form, but they don't necessarily always do that. So the spread of cancer, there seems to be some like direction. It's not like it's going to just spread anywhere and everywhere. There has to be a convenience issue. So if there is a primary cancer that is in the bowel, so this is your GI tract, this is your large intestine, and so then it's been found that, you know, there has to be a convenient location, and a secondary cancer can be found later on in the liver. Okay, so it may not be necessary to the right next adjacent tissue, but it would have been to a comfortable location somewhere, somewhere where it is able to grow. And so that's the secondary cancer that has spread. And so some examples of other primary cancers and where they spread to, it seems that there is some kind of a pattern, and that pattern is that they don't just go everywhere and anywhere. These cells will spread into some kind of convenient locations, like I said. So the blue location, so here it is, let's go down here, let's look at this pancreatic cancer. We have not talked much about pancreatic cancer at all, but pancreatic cancer, they have some favorite places that they like to spread, and so it will spread in the liver and it can go into the lung. That's the area that it shows. Additionally, let me let this person in. Do we admit? So additionally, if there is a lung cancer, as you see here, maybe I'll just draw a circle. If there is a lung cancer here, then it can spread into the bone and it can go to the brain. So that's kind of like a favored location. And if there is skin melanoma, again, that skin melanoma that is shown here somewhere on the shoulder here, then that also can go to the brain. So the chances are that it goes, preferably, can go to the brain. And one last one here that I wanted to show you is colorectal cancer down here. That's in the colon. That, as we just talked about a couple of minutes ago, that can go into the liver. So these are kind of like the tendencies that have been understood. And I did not say that lung cancer only goes to the brain, but it can go to the adrenal gland, and adrenal gland is right over the kidney. It's a little bean-like thing right over the kidney. And so those are some of the favorite locations. Let's see. So, how does it spread? So it can spread the cancer cells. They have to literally travel. They have to really go, but they're not going to go through just the tissues. They will not go through other cells, but they can really go through really these two ways. They go through the blood or they go through the lymphatic system. And so if it has to go through the blood, then first of all, it has to separate, break away from that primary cancer, which is a lot of work. It's a lot of effort with the tumor microenvironment, as you saw. And it has to be able to travel through the blood. It has to survive through the blood. And sometimes it will therefore go into the blood vessels, the smaller capillaries. Some of them really go into the smaller capillaries. They're not just going in through major. So going to the smaller capillaries means that it can go into some smaller locations where it might get stuck, and that's what it's really saying, that it might get stuck in there. So let's look at that travels by the which travels by blood circulation. So if it is going through the blood circulation, then and these are not fairly small blood. Whoops, sorry. There we go. Fairly small blood vessels. Okay. So yes, it can first. I'm not going to show you the whole circulation of the body. I could, but it's not necessary. But once they get into some of the smaller blood vessels and they're traveling fairly freely, then they're called as CTCs. CTCs are circulating tumor cells. CTCs. And these CTCs, they can, again, that language that I use, they can get stuck somewhere in not a small blood vessel, but a very small blood vessel. Okay, and that is a very thin blood vessel, such as a capillary. And our capillaries, they are meant to really allow red blood cells to travel and squeeze through very finely, but they're not really meant for larger cells. So the cancer cell has to be able to find a way to kind of like move through the wall of the capillary and into the tissue, or just kind of like try to go grow in here. So first of all, it'll be stuck, and then once it's stuck, it might be able to figure a way to even multiply in here, and form a tumor right here. Okay. And then try to evade and go to the other cells. And if you remember, if you see this blood vessel, I use this language before also, endothelial cells. Okay. Most cancers are epithelial, but these are endothelial here, you see. And so these are endothelial cells, endothelial cells. So this is the lining of the blood vessel, endothelial cells everywhere with the nucleus. You can see those nucleus, and in the capillary wall. So they can multiply to form tumors. They can give pressure there. They can try to get out of it, but they do get stuck, and then that's where they try to um, try to evade this, try to grow out of there if they cannot move any place else. And so the second one is through the lymphatic system. And this is where I thought there should be a diagram for you uh, for the um, for the lymph nodes. And so, the lymphatic system, it's really a network of glands, lymph nodes, glands that are like filters. There's a nice language. There are filters, and they are locations for lymphocytes, other white blood cells. And whenever you have an infection, your lymph glands can get a little swollen because they're doing all the fighting, and all the dead bacteria, they go there. So they are really filters, and those you have lymph glands in the armpits. These are just the locations to show you. Okay, somewhere up here, there's this groin area, and these are the major locations. And so the others that they're showing here, these are also all of these. Look at all of these. They're in the chest, right? And so, and they're just flowing here. So the node is here. This is a node near the knees up here, but that is just for for the cells to flow. So the cancer cells, can they go into the lymph vessels? Again, they had to squeeze through. They figured out how to squeeze through, and then they travel to the nearby lymph glands, and then they form tumors inside the lymph nodes. And yesterday you did learn that it can either be in the node or it's not in the node. So remember there were scores for that. So it's either nodal or it's not nodal. So once it's in the node, then it will just get bigger right there. So what's the word metastasis mean? Metastasis or metastasis, call it any way you want. Metastasis or metastasis is really that process. It's the process, the whole process by which the full process by which these cells, the tumor cells or the cancer cells, will be able to invade. So this is nice formal language here. They will be able to invade, and then they will be able to travel or migrate to the other parts of the body. That whole process is called as metastasis. So the cancer can spread by the process of metastasis. So the spread of these cells, if it goes in somewhere, if they have to spread throughout the body, again, when they grow, these cells will multiply, will grow, and then they can cause physical obstruction. Where can they cause physical obstruction? If they grow as a tumor in certain locations, there's no more room to grow. These cells, these cancer cells that will go to the body, will also compete with normal cells. For what? For all the nutrients. Okay, all the nutrients that traveling, and the hormones, etc., that are traveling through the blood, and of course oxygen. They will compete for that. However, we saw yesterday there's a lot of oxygen, but it's not using all that oxygen. Remember the Warburg effect? And of course, they will now interfere with any organ that they go to. They will interfere with the organ function. So not only will these cells not do their own job, but they will also go and interfere with other organ functions. So this whole process, to understand this whole process, what it is, that's called as metastasis. Excuse me. So the bloodstream, the blood flow being the major means of long distance transport, so not really the lymph, but the blood being the first and ideal way to transport. So all those organs really that are really on the road, the one that are near the highway, all the organs that are near that. So those are going to be typically the main sites. And so therefore it's kind of like there is a preference. If the blood circulation goes straight somewhere, then it is a preference to for the cells to just go through that. And that is a reason to understand why metastasis occurs in that particular location. So again, if it's in the GI tract, it's easily possible that it will go to the liver for that particular reason. And so there is this theory that that why does a metastasis take place? The bottom line, the question I'm asking or saying is that the cells, these cancer cells, where do they want to go and form a secondary tumor? Where do they want to go to form a secondary cancer? Where should they go? Where do they want to go? And it seems that there has to be a convenience to them. They just cannot go everywhere and anywhere, but they would like to be able to start, and they would like to be able to. It's just like a seed that gets the right kind of soil. So the seed soil theory, this is an old theory. The seed soil theory means that the cancer cells are the seeds, and there has to be some kind of optimal environment called as which is the soil. So the tissue that they go to, so that means the tissue that these cancer cells go to through the blood, they have to have some kind of convenience. Just because it's nearby and in close proximity doesn't mean always that there will be metastasis. There has to be convenient conditions of the right pH, of the right amount of secretions, etc., that will make this particular that make it possible. And so not only does the cancer cell have to go and be like find the nice garden for the seeds to sprout, not only that, but then before that, the primary tumor can also kind of like wait, and the primary tumor, the primary cancer cells, can release can release certain factors. When I say factors, I'm saying that they can release certain substances. They can secrete them out. They can release them and and keep sending them to these different locations. And all they're trying to do is preparing for the arrival, you know? So okay, isn't that kind of smart? So it's like a preparation for the arrival of those tumor cells. So that means it can release secretions such that maybe the when they land there, they have they have certain substances that make it comfortable for them to attach once they've gone there. So all kinds of things. That's just one example. So it's called as the pre-metastatic niche. This is the word is niche. Pre-metastatic niche. I want to create a comfortable home and a comfortable location beforehand. So I keep on sending, I means the cancer cell keeps on sending little substances, and there's some locations that might make it really favorable. And so that is it. So what you just learned right now, it is not only does the tissue have to have the optimal environment, if it goes through the blood, it has to go somewhere where there's optimal environment, but the cancer cell can create that optimal environment. That's what this is saying. That the cancer cell will find ways to make that optimal environment. So it's like getting ready. It's like a griho pravesh, putting things there before you go, and then settle down. So, I talked too much. It was already here. All right. So, seed and soil theory. So, all these interactions, that's a nice word. So, all of these interactions needs to take place. And so, that was the reason why, even though the cancer sells out these substances, some of the locations, okay, some of the organs, location means organs, have a preference. So, there's a preference for certain cancer. So the figure that I showed you up in the beginning, as with those arrows of blue and yellow, if you don't mind my going back to that, these particular arrows, this was really a kind of like a preference based on the primary tumor to prime itself and to go in those locations. So so it goes to those preferentially to those places. And so which are those examples? So one example I have for you here is breast cancer often goes to the bones and lungs. So women with breast cancer, they have to constantly make sure that the cancer doesn't settle down into the bones and lungs. And so it's all because those cells, they have been able to find suitable ground, find suitable soil where those breast cancer cells grow. So those same breast cancer cells have to be like comfortable to be growing in there. And so that is so if the person, you know, didn't die of the breast cancer, but there was some metastasis and it went to the lungs, then the person might just really die because it went to the lungs, you know, because of this metastasis. So it's of prime importance to prevent that metastasis, especially, you know, in this situation. All right, so the soil, which is that new organ or the new place where it will go, has all includes factors like the tumor microenvironment. So the microenvironment, it is not the tumor, but it's just a microenvironment of the new location, and that microenvironment are things like the immune system. Okay, we're not going to talk about the details of the immune systems, but you know that immune system is composed of lymphocytes, of various other white blood cells that support and prevent infections, etc. And the other things So the microenvironment has the immune system. So the cancer cell has to make sure that it can trick the immune system, that it can trick that microenvironment, and turn it into a friend and say, so that it can be able to grow at that region. And secondly, also that there are adhesion molecules. All right. The cancer cell needs to have all these various adhesion. Adhesion means to stick, to stick together. So adhesion molecules that it needs for able to be able to go and locate and stay near that place and not be destroyed. So it has to be able to stick, or it has to be able to adhere to the new tissue, to the new location, not tissue, but just location, through the blood vessel, to the location, in order to survive. And so all those factors, the cancer cell will either have to create it in its favor. Maybe it would like to have certain adhesion molecules. It'll make them. It'll start to tell that location to help it to survive. So it's like you moving to a different location and everybody helps you to set your new house. It's kind of like that. But here, it's the cancer cell which is telling everyone to, or forcing them to help. And so that's the same picture that explains that. Or um, now recall the Warburg effect. Recall the Warburg effect, which is the metabolic change in those cancer cells, and they consume a high amount of glucose. They want to use the glucose. They will utilize it. They will break it down, but they'll do that without the oxygen. Okay, there is oxygen, but they will just not use it. And so I just want you to remember that, because that kind of combines everything in our um, next slide here, which is a little bit of explanation of how that metastatic, you know, tumors that might have gone somewhere, how do you detect them today? So I just wanted to bring in some technology here as well. And so this is an example of non-Hodgkin's lymphoma. Where is the lymphoma? Well, it is in the hold on. So where is the lymphoma? Of course, it's in the lymph nodes. So in stage one, in stage one of the disease, it's localized. Where is it localized? So I just had that red arrow for you. It's localized here. You see that it's in the lymph nodes, all right? And it's one lymph node in one location, all right? So it's in one location, or one place, or one single organ. And then in stage two, the lymph node, it has traveled through the lymphatic vessels, and it is in a second region, in but also in the lymph node. Okay. And in stage three, it might have traveled a little further down. It might have further, traveled a little bit further in other regions. And until, you see the liver here, the liver was not affected. But then later on, it affects the liver. There you go. And it affects the bone. Okay. So that's because not necessarily that it went through the lymph nodes, not necessarily that it went through the lymphatic vessels, but maybe from the lymph, there is this, it might have gone through the blood vessel, and so from the blood vessel, it might have traveled to the liver and to the bone. So the cells, they are able to travel because the lymph and the blood vessels are kind of like very close together. And so then it leads to widespread, and so that would be your stage four. So that would be non-Hodgkin's lymphoma, which which has a survival rate about 65 to 70%. And so, how would we detect all of this? If they're circulating, these are tumor cells that are circulating, it is not circulating in the blood, but this one is in the lymph node, then we can perform these certain image scans. So we can have MRI, we can have CT scans, we can have also a PET scan, okay? And so this scans all the body tissues. And so I just wanted to show a movie of this, and it is fairly a little bit of kind of like a longer movie, like as in like 4 minutes. But I just wanted to bring in technologies, which technologies and how the MRI, it's really more focused on MRI, how the MRI and PET are different. And this just wanted to tell you that the glucose is kind of like involved here. All right, and so once we've seen that movie, I just wanted to show you that we will take a look at this image here and we will just kind of like analyze it once we have examined that little small short movie. So let me see if I can get this going. Seems to work. Okay, and what do I have to do? I just have to go to the 1 minute.
MRI and PET scans are two commonly used diagnostic imaging. MRI and PET scans are two commonly used diagnostic imaging tools that play a crucial role in the diagnosis and monitoring of various medical conditions, including cancer. Both MRI and PET scans produce images of the inside of the body, which can provide valuable information about the structure and function of internal organs, tissues, and bones. However, while both tests can be useful, they have some significant differences that patients should understand when considering which test is best for their imaging needs. One of the main differences between MRI and PET scans is the imaging technique used to produce the images. Magnetic resonance imaging uses a strong magnetic field and radio frequency waves to produce detailed images of internal structures, while positron emission tomography uses a small amount of radioactive material and a special camera to detect changes in metabolism and blood flow. MRI scans work by aligning hydrogen atoms in the body with the magnetic field and then using radio waves to disrupt this alignment. As the hydrogen atoms return to their normal alignment, they emit signals that are picked up by the MRI machine and used to create detailed images of the body. PET scans, on the other hand, work by introducing a small amount of radioactive material into the body. This material is typically injected into the patient's bloodstream and travels to the target area where it emits positrons. When positrons collide with an electron, they produce gamma rays that can be detected by the PET camera. This scan data is used to create images that reveal metabolic activity and help identify areas where cells are using more energy. Another significant difference between MRI and PET scans is the length of the scan. MRI scans typically take longer than PET scans, often 30 to 60 minutes for an MRI compared to 15 to 30 minutes for a PET scan. This can be a factor for patients who have limited time or who may experience anxiety or claustrophobia during scans. A key difference between MRI and PET scans is the amount of radiation exposure involved. PET scans use a small amount of radioactive material, which can expose patients to low levels of radiation. This can be a concern for some patients, especially those who need repeated scans. On the other hand, MRI does not use ionizing radiation, making it a safer imaging option for patients who need repeated scans or who are concerned about exposure to radiation. Both MRI and PET scans can provide valuable information about the inside of the body, but they provide different types of information. MRI provides detailed images of anatomy and tissue structure, which can help diagnose and monitor conditions such as tumors, inflammation, injury, or degenerative conditions. PET, on the other hand, provides functional images that reveal metabolic activity and help identify areas of the body where cells are using more energy. This can be useful in the diagnosis and monitoring of conditions such as cancer. In the case of cancer, MRI can provide high contrast images of tissues and affected areas, which can help doctors determine the size, location, and extent of the tumor. MRI can also help distinguish between benign and malignant tumors based on the characteristics of the tissue. PET scans, on the other hand, provide functional images by administering a glucose-based PET contrast agent that travels through the bloodstream to reveal areas of increased metabolic activity and blood flow. This imaging technique takes advantage of cancerous cells' reliance on glucose in the bloodstream for energy. PET scans can help healthcare providers determine the stage of the cancer, identify areas of the body that may be affected by metastasis, and monitor the effectiveness of treatments. Thank you for watching.
So I hope that was a little useful. So essentially what I wanted to really show you here was that was the fact that you can use the PET scan and combine the whole concept of the Warburg effect, um, uh, and look at those tissues that are utilizing more glucose. That was really the whole idea for introducing the PET scan. And so I want to show you this slide now. And this is a slide. And because we're talking about images, but not necessarily always, we have to look at cells. And so this is a combined, it's a fusion image, but both things were done. And this is a CT scan. All right. And it's a CT scan, but it is done with PET. So, I mean, PET was also there. And then, so they're both, it's a fusion image um, of the both. So all the body tissues, uh, for the same example that I just gave you for non-Hodgkin's lymphoma. Okay, so all the body tissues here are shown in gray and in blue. And but glucose was given, and the way that the glucose, radioactive glucose, is given, it's fluorodeoxyglucose. I guess that's probably the standard. And it shows up, in various tissues as yellow. Okay, so you have yellow here, yellow here. That means those are probably locations where this glucose is being utilized. It's breaking down. So I imagine that it's probably just immediately being broken, and when it breaks down a lot, then it gives you this particular yellow like so. So high glucose uptake is FDG indicates, uptake, high glucose uptake is indicating all that glucose, big glucose use, but with the Warburg effect. All right. So here, so you have yellow. Where do you have yellow? The yellow accumulation is in these locations in the abdominal region, but there's also yellow in the brain. And they are saying that in this picture that the yellow in the brain is typically normal. That is kind of expected, but the other spots in the abdominal region, that's where the lymphoma, the cancer from the lymphoma has spread. So that's the spread of non-Hodgkin's lymphoma. It's not just there in the lymph. So these are lymph locations here. These would be the lymph locations here, but it has already spread to the other locations. And so I have a question here. I just don't know which one is the original and which is the primary and which is the secondary. But that's not a question we can answer today. So it's a little hard to understand, which one's primary and which one's secondary here. But the bottom line is it's not just in one place. It's in a bunch of different locations in the abdomen here. So that was the utilization of Warburg effect from yesterday, as we said, and for lymphoma, non-Hodgkin's lymphoma, as you know now, and the fact that you can use that for tomography. So metabolism is used for positron emission tomography. Okay, let's go to the next slide. And so what are the total number of kind of events here? So the whole there has to be a bunch of, it's a multi-step process. There's a whole cascade of events. And so we will look at some of these in quickly, and we will look at other things in a little bit more detail. And this is a picture that you saw yesterday, or the day before. And so the multiple steps are six. There are six steps to the metastatic cascades. Okay, I hope you're paying attention because I'm going to ask you these questions on the last day. Alrighty. So the first step, so this is your first one here, is this. We'll just go through this a little quickly. Let me see if I don't have a bigger picture. This is the one. So here's your primary. So now you know the word primary. So your primary tumor formation. When we went through this, but now we'll look at it slightly differently. So localized invasion. Localized invasion is the step number. Is step number one, and it seems that this one, this is a lot of things have to happen here. A lot of activities have to happen here before intravasation can occur. So what is the activities? Here, localized invasion means that the cells, they are just getting bigger. They have formed a tumor, but they have not entered the blood vessel yet. They might want to enter the blood vessel, and the cells have to do a lot of effort in order to do that, those cancer cells. So we will look at this a little bit more in detail. That's what it really is about, localized invasion. So, and then what leads to that second step? The second step is intravasation. Intravasation. Vasation means in the vessels, in the blood vessels, and intra means going into those blood vessels. Okay, so um, sorry. Just give me a second. I'm just trying to see other faces. So intravasation is an important step, but it simply means that the cells have entered the blood vessel, and here now the cells, those cells, they have to interact with all of these other members. There's other people, not other people, but there's other members here, platelets, lymphocytes, all the other blood components. It has to learn how to deal with them, right? So that's intravasation once it's gone, once it's inside. So the third one is transport, and for transport, it has to go through the major, after wherever the blood is, it eventually has to go through the heart. So it will go out through the aorta here, and it can travel through the whole different body, so that it can go to the all parts of the body from there. So that's transport, and that's plain through blood circulation. That's what we're talking about now, not from lymph anymore. And then where does it go? From there, it will now go into all those different locations, those small capillaries. So it goes to the microvessels. And so once it's in those microvessels, that's where it can get stuck. So arrest, that's the word, okay? So it can get stuck in those microvessels, but through the blood, etc., it can go to various parts of the body. It can travel, and it can decide which organs it wants to go to, which ones it's going to feel more comfortable. Like I said, it has to find a comfortable location. So let's say that it went to near the bone through the blood vessel. It's near the bone. It is near the bone, let's say, through the tissue that is near the brain, let's say, or it is near the lungs. So then the next process would be extravasation. Extravasation means coming out or outside, and vasation refers to the vessel, so coming outside of the vessel. And I want you to take a look at this very carefully. See that extravasation, what's happening here is that the cells are modifying their shape in order to come out of the blood vessel, this is the blood vessel, coming out of the blood vessel and into the tissues. And so now that it's going to come out of the blood vessel, it needs to settle down somewhere near the blood vessel, or a little further from the blood vessel, etc. And it needs to form a micrometastasis. A micrometastasis means it takes a while for all most of the cells, they don't really live. Even if they go, most of these cells, even if they go and settle down somewhere, they don't form micrometastasis so easily. Okay, it's just not always possible. So some leaky cell is going to form a micrometastasis. It's not a suitable environment for everyone, so that's why. And then colonization. And in colonization, they are growing bigger. And how does that happen? Colonization requires blood vessels, etc. And so it's a macro metastasis now. So it's now bigger and it knows how to survive. And I will show you one or two videos about that colonization. So now what we're going to do is really, this red arrow is just showing here that we are going to talk about some of these others. Some of them will be videos, and what we're going to spend some time as to what is it that allows these cancer cells to really go into the blood vessel. What are they doing during this whole time? What are they manipulating? Who are they manipulating? What is it? Is it doing it to the tumor environment? In order for metastasis to take place. So let's look at that. So localized invasion, this is the same thing again. And today, for this one, I might need for you to, right now, to learn a little bit about some molecular biology. So some names I'm going to throw out to you. And these are, whenever I use the word molecule, I don't mean a molecule like a physics molecule. I'm meaning a molecule as in a protein, as in a substance that the cell makes. Okay, so it's a protein that the cell makes, and we just call them molecules. Everything is a molecule, but we just use the word more. So in order for invasion to take place, for them to invade the local environment, the cells have to break free. Okay, so they have to break free from their location. They have to come out of that basement membrane. Remember, epithelial tissue basement membrane. And so they have to break from all those constraints that link them to the other cells. Okay, it links them to the other cells. They have to be able to uh, break away from those cell adhesion molecules. So they have to manipulate the cell adhesion molecules. And which are these cell adhesion molecules? Number one is E-cadherin. E stands for epithelial, and cadherin. All right, cadherin because it's adheres, it's help for adhesion, adhering. And Ca is because it needs calcium. So it's calcium-based adherence. And another one is called as catenin. So you don't need to know all of these names, but you'll see these a bit more often, so therefore they're here. So all they need to do is now they need to be able to manipulate these cadherin and catenin. Okay, and so what is one of the best characterized alterations? What can it do? What can the cell do? Maybe it can cause a loss of E-cadherin, loss of that cadherin that is required for adhesion. The carcinoma cells will be able to, maybe they will acquire they can acquire some changes such that there is a E-cadherin is very little. And so if there's not much adhesion, then good for the cancer cell. So again, what is E-cadherin? Just to give you that background, it is important for epithelial cell to cell adhesion. Cells have to stick together. They need brotherly and sisterly love, like I always say. And so they form these junctions. They adhere. Everything is, the word is adherence. Adherence junctions, where next to each other, next to the individual cells. And so that is your E-cadherin. And so it might need to manipulate that. Okay, so all I'm just saying is that the carcinoma cells, okay, the carcinoma cells or the cancer cells, the best example, need to manipulate and make E-cadherin inactive, or maybe just lose the capability for E-cadherin to function. So no adhesion. And what does adhesion mean? So I'm going to draw this here. That's your basement membrane. And remember, these are the cells. And so there's some extra cells here. And so all of them, they just want to break open from each other and break open from these other cells. So let's say this is the cancer cell. This is the cancer cell. They want to break free from whom? Break free from this one. They also want to break free from the basement membrane. So they want to break free from the neighbors. They want to break free from these neighbors, but they also want to break free from the basement membrane. So E-cadherin is between these two cells. So it seems that, some cells are we don't understand this yet, but some cells play like have the role of a leader. So I wrote, following the leader cells. Maybe there's some cells like to become a leader. And so this is an experiment diagram that is we're showing you, where cells have been, stained or labeled fluorescent. So what are leader cells? It seems that leader cells take that special role of being the first to go out, and try to leave the other cells and find a way to go to the blood vessel, or to invade the other tissues. Okay, so this is a primary breast cancer tumor. So this picture here, and so which is the cells? So there are green cells and then there are pink cells, and so these green cells, these are all the breast cancer. These are all the breast cancer cells, BC. And you see these pink cells here, these skinny, skinny pink cells. You see, do they look like, well, you can't really see the shape of all the cells here, but here's one shape, and there's a nucleus. So you can see some of these cells. They have a nucleus. And so some of these cells, and look at the pink one here. See that pink one? See these pink? They're very skinny. They're not the same shape. They look very different. And so they have been stained, and that's how they were found. So these are tumor cells, but the pink cells are the leader cells. And the leader cells, as you can see, these pink cells here, they are beginning to break off from the main body. They're bringing to break off from the main body of the tumor cells, and they are progressing in this direction. They are progressing outward. And so these breast cancer cells are able to come away from the primary tumor. Okay, so it's coming out from the primary tumor and it's trying to invade and colonize the other tissues. Alrighty, so it doesn't matter that it did not go to the blood vessel yet, but it is trying to go in and find ways to go to the other tissues. So scientists have found out that in these breast cancer cells, all right, how do they how is it possible that some of these cells, they kind of like look different and they are the leader cells? So if these are leader cells, scientists have found that they are genetically different and they are behaviorally different. So some cancer cells will modify themselves, and they will turn into leader cells such that they can be slightly different from the main body of the tumor. Okay, so now you see that cancer cells are also kind of heterogeneous. They have different properties. Some of them are bigger, and some of them have seems to be completely different morphology. And so I'm thinking the reason why I brought this slide in here for you, I'm thinking that maybe if there are indeed, if there are indeed leader cells, and they are genetically and behaviorally different, then maybe, and I can see that they're also a little morphologically slightly different, then maybe you, as the computer science, the ML, and the AI person, you can go and consider the ways by which you can study these, find slides for these, and be able to see if there are in existing slides there are some cells that have this kind of morphology, and they can be identified, and then they can be screened, and then it can be detected later on, and that you can use this for later things. So this is an idea thrown out to you, and this is to you to see if that leader cell has a sufficient morphology difference for machine learning to probably identify them and classify them separately. And so if they are caught early on, a person's and it may not be in the tissue, maybe this could be if it's a breast tissue, can we find these by fine aspiration, fine needle aspirations? Can we Can we the least amount of invasiveness. But the bottom line I'm telling you is that if there are enough data sets for these, if you have enough data set, then you might want to look for them, and you might want to take it to the next step. So, and I'm going to go for a little faster on some of these, which which are not so much for morphology based and don't involve much of cytology right now, but I still, so some places will go fast and some places will go slow. So the next one is matrix metalloproteinases, MMPs. These are enzymes, and these are enzymes that will go, and typically, just to give you a reminder, if these are the cells in a tissue, then outside is the ECM. Outside is the ECM. So everything outside is here is the ECM. And so what is the ECM? Cells are secreting stuff. Cells are secreting stuff, and they're maybe secreting it out into ECM. So the ECM also has collagen, remember from yesterday? And so the ECM has collagen, so that these cells cannot go and break away also. These cells, everybody's held together. The cells just cannot go and break away. They just won't happen because there are all these different kind of proteins besides collagen that need to be, that the cell just cannot break away. So the tumor cell needs to be smart, and the tumor cell, okay, needs to be smart, and the tumor cell is thinking, well, if I can just, so if this is a tumor cell, and it is thinking, well, here's all of this, collagen, and here's all these fibers, okay, again, so let's say these are all this ECM. How can I break everything in the ECM? So this is ECM. It has all kinds of fibers. So it is probably wondering, how can I secrete it? So this cell, the tumor cell, so these fibers can be cut down by this enzyme called as MMPs. So MMPs are proteases. They break down these proteases, and they degrade that ECM. They degrade everything in the ECM. So if they degrade the ECM, this gets wiped out. If they degrade this one, this gets wiped out. If they degrade this collagen, and not just collagen, but there are so many other molecules. And so if it degrades all of this and makes it all loose and thinner, then this cell can then be released. So so a tumor cell would like to invade itself into the surrounding tissue by using MMP, and the way that it might do that, the in some tumors, these carcinoma cells, they make their own MMP. So they will make MMP. They will make their own MMP. Generally, it's supposed to make a little bit when needed, but this somehow they just manipulate, and they'll keep making their own MMPs. You don't have to memorize the whole thing, just call it MMP. So they will make the own MMP. So cancer cells will make the MP, and so what will happen is this is all get wiped away. Look at this, it's all wiped out, and now the cell can easily break out. This cancer cell can break out. While in other cancers, what's happening is that in some cancers and in others different, every tumor is kind of different, the macrophages, which are neighboring cells, the mast cells, which are neighboring cells, the fibroblasts, connective tissue, that are all surrounding in the tumor in microenvironment called as the stroma, they secrete it. So the cancer cell, let me see if I diagram. So the cancer cell tells, so here's the cancer cell again. Let me draw the cancer cell here again. So more tumor cells. And so all of the macrophage, so what does a macrophage look like? The macrophage looks like this. These macrophage within the tissues, they're talking to each other, and the cancer cell kind of like tells the macrophage, all right, it tells the macrophage, can you do me a favor and can you make some MMPs? And then the macrophage will make the MMP, and it will degrade the collagen. So here it is. The macrophage will secrete it. The macrophage will secrete, and so let me show you dots here. So doing the dirty work for the cancer cells. And then degraded. So macrophages are recruited. A cancer cell is so smart that it'll tell the macrophage, come one and come help me, please. And then the macrophages will do that. Okay. And so the fibroblasts, let's just ignore the mast cells, the fibroblasts also. What are fibroblasts? Remember, the fibroblasts are those skinny cells that are that make the ECM. They make all of this ECM. They make them surroundings. Okay. So MMPs. Okay. So now we come to a part, like I said, remember I said I'm just going to go back a bit, and I said that we're going to spend more time, a little bit more time on this whole thing. Here, there's just so much happening here. So in localized invasion, we still haven't left it yet. In localized invasion, if the cancer cell is going to decide to leave and sneak through, it's not so easy. There's so much ECM, there's so many other cells, tissues everywhere. Then it needs to become a bit skinny, and it needs to be very, very pliable, and it needs to become a little pliable and moldable, and it needs to be skinny so that it can be like a child who's skinny, who can just go and squeeze in through small locations. And this whole big phenomenon that I'm talking about is called as the epithelial mesenchymal transition. Epithelial means that it is an epithelial cell. As you know, all these cells are, most cancer cells are all epithelial. Okay, we're not talking about the lymphoma right now. So if they are a solid tumor, it's epithelial, and they change into a morphology. Their morphology changes into, or their morphology, more than their morphology, their characteristics change such that they are called as mesenchymal. So they have the properties of a cell that is skinnier. I mean, it's just an example that a word that should not be used, but I'm using it. They are skinnier. They have a much more capability to to sneak through blood vessels, etc., and just travel faster. So that is called as the mesenchymal properties. And so they acquired this ability. And so I'm going to give you a diagram in a bit. Just hold on. And so this is a transition, and this transition is called as the EMT. All right, epithelial mesenchymal transition. So it's a major mechanism by which these neoplastic epithelial cells will acquire the ability to invade. Again, they've they're changing their shape, they're changing their morphology, and they will invade. And they will invade means they will sneak through, and they will resist apoptosis. They refuse to die, and they will just go and pass. They will go and easily spread, disseminate into various tissues. So, so what does this involve? You have a sheet of closely connected epithelial cells. So here are all of these epithelial cells, okay? And of course, they're slightly forming a tumor. They're growing on top of each other, and they are going to all convert into cells that look like this. So instead of being square, they're now going to look like this. So this is already, it's not like it doesn't happen in nature. It's already very common in embryogenesis, in wound healing. Whenever you cut yourself, you need cells to quickly travel. They need to make more new cells, and that gash that you have, that wound, it gets filled. All right. So, so this is happening in these cancer cells. Okay. And so this is how it looks like. So this is your basement membrane. Here is your basement membrane. All the cells, you see those little blue dots here? Those blue dots, those are adhesion molecules. They're like little staplers. They're holding the cells in the place to the basement membrane. And do you see these other staplers? These are junctions. Okay, so these are junctions, and they're like little staplers. They staple the cells next to each other. So this is very strong. If I try to push and try to punch this, they will not break easily. So that's how strong our cells are, held together in sheets. So this is in a like sheet configuration. That's normal. Okay. And in EMT transition, in an epithelial mesenchymal transition, mesenchymal cells, we're saying that they have this shape. And you can see this is a shape that will help it go through. So this is your ECM. You see this ECM, there's all this collagen here, this gray stuff. And now it will be able to travel from this in between that collagen. So if it can also cut the collagen, but it can also travel. And so this is a big study. So uh, epithelial cells have cell polarity. Cell polarity means that this is the top of the cell and this is the bottom of the cell. They're polar. So this is the bottom of the cell. Okay. Generally, there's no such thing. Here, there's no top bottom. There's no cell polarity. There, I don't see any here. I don't see any top and I don't see any bottom. They all look alike. So big loss of characteristics. So now you see that this is morphologically slightly fairly different. And cell adhesion, they're all stapled together like I just showed you. They are held to each other and they're held to the ECM. The ECM is down here and this is the basement membrane. So now what's going to happen in EMT is there's a big loss of cell adhesion. This is the big advantage. They don't have to stick to each other. So it will break all those staplers. The cancer cell will know how to break the junctions. And so it uses enzymes to cut away. It will use enzymes to cut away from here. It will use enzymes to make this collagen even smaller and cut away from here. Then epithelial cells are stationary. They stay in one place. They're all your tissues, but these are able to completely migrate. And they are going through tissues, going in through blood. And so they will invade. They can even squeeze further. They can squeeze further and go locations. And that word I said, they have cadherin. So here you can see that they are well attached. So there's high level of cadherin. Cadherin is a protein that helps for adhesion, for adherence. And so they will have high level of e-cadherin. And it's good to know for you as well. Whereas these cells, they don't have all that cadherin. They just don't need them. So they have low level of cadherin. And by the way, where is the cadherin present? Just to show you, the cadherin is present in these little junctions. See those junctions? That's where there's cadherin. This is where there's cadherin. This is cadherin here. So it contains this molecule called as cadherin. It's a very special protein. And so it kind of like degrades the cadherin and it doesn't even make it. All right? It doesn't even make it here anymore. So if you take a look at these cells and if you stain them, they just don't have it. So I'm just wiping it away. They just decided not to have any cadherin. So low level of cadherin. So low amounts of cadherin, high amounts of cadherin in epithelial cells. And you can just ignore the N-cadherin for now. Okay. So those are your major properties of EMT transition. And so that's what we're going to focus on, honestly, today. So this is another diagram. You don't have to pay too much attention to this, but it's just to tell you that this is the epithelial-like morphology, and this is the mesenchymal like morphology. Okay, so mesenchymal cells. Now, mind you, those are not the leader cells. I showed you leader cells. That's kind of a little bit even further different. Okay, so we're still trying to figure that out. So those are not exactly leader cells, if that was your question. And so these are all these very regulators, these EMT regulators. There's these special genes that we now know. These genes are, this DNA or they make proteins. These are all slugs, snail, jeb, so these are different things. We don't need to talk about them today. But epithelial markers, this is the language I want you to look on. Epithelial markers are inhibited or down, and mesenchymal markers are those that have increased. And so just for you to understand that N-cadherin, even though it doesn't, I said to ignore it, you have a lot of N-cadherin here, that's fine. You have more E-cadherin here. And so another one that I want you to look at is vimentin. If it's a mesenchymal cell, it will have more of a protein called as vimentin. So what did we just say? So we have vimentin here. Okay, this is good for you to know. Vimentin here, and here you have more cadherin here on this side. Okay, and what else? I think that's pretty much it. Okay. And some of the mesenchymal is that there is still collagen. Look at this. There is still collagen, but it is type 1 collagen. Type 1 collagen, but in epithelial, they have type 4 collagen. So type 4 collagen makes a sheet, it helps make a sheet and crisscross and is very tight. Okay. And type 1 collagen is a little bit loose and it is more forgiving. And so now these mesenchymal cells, they have acquired properties of invasion. It will help it to metastase. They have properties, I've not explained how, but they can avoid cell death. So what's the problem there? We have now drug resistance. That's a big, big problem. So I have not explained how they avoid apoptosis, but they will not die. So give drugs, they will not die. Okay. And worst of all, that these mesenchymal cells, they have some stem cell like properties. Remember, I said what are stem cell like properties? Making another clone of you. And so they can now form cancer stem cells. And so a cancer stem cell means it's going to be there. It'll just create another one of itself. Okay, so let's talk some more. You as the computer science uh, students, can we detect? So I'm asking you, can we detect these cells that are going through EMT? EMT is like a phase. You were epithelial now, and you converted to mesenchymal. So it's like a phase. So can we? Is there any way to find them? So yes, we can do cytology. We can do some kind of cytology, but it has to be done. That cytology has to be done with antibodies. So it is immunocytochemistry. Immunocytochemistry means that you have to label the cells. You have to find the cells with antibodies and give them a color, give them a stain, so you can detect them. Or the other one is immunofluorescence. Immuno means again you have to use antibodies, commercial antibodies from the market, nothing from the body, from the market, and those antibodies, and along with a dark background and fluorescence with UV light, they give you that bright green color. They are used to identify the EMT and the ME. Sorry, I'm going to ignore this right now because I have not talked about it. Therefore, giving you the EMT cells. But where are we doing this? We're doing this in cultured cells in the lab so far and in clinical cytology samples. There you go. So it's being done. So that means this is like extra processing. It's not as simple as your H&E stain. It is not as simple and cheap as your or inexpensive as your pap smear like we talked about. This is more expensive. So this is more work also. So anything to do with immuno is just more work, and it just needs a slightly better infrastructure for immunofluorescence, but still doable. And so what do people find? What can you find when you look for E and M, so epithelial? Then what you're looking for is in your observations by doing immunocytochemistry or immunofluorescence, any one of them. The expression level. Expression means how much of it is made inside a cell, how much of a protein isn't made. So just want to say that because these are sometimes hard to see, therefore looking at these markers is the best way. So looking at these markers, these proteins, is the best way to do it. So look for cadherin, look for vimentin. All right, so expression level of these markers, the marker means E-cadherin and cytokeratin. And that's all you need to know. E-cadherin and cytokeratin. Cytokeratin is inside the cell. E-cadherin is like those staplers I showed you. So you look for these, or you look for mesenchymal markers. So these are epithelial markers. This is for epithelial marker, and or maybe the cell has mesenchymal traits. And so you look for this one called as vimentin and N-cadherin. Okay, even though I said ignored, I guess it's more important. So N-cadherin and um, so N-cadherin, typically the N always stands for neuronal, because probably it's found in neuronal a lot, but it's also found in every other cell as well. So if the cell has more vimentin and it shows a certain color, then it's a mesenchymal cell, and that's like evidence. And so a decrease in epithelial markers, okay, a decrease in epithelial markers and an increase in mesenchymal markers means that EMT is taking place. Okay, so I'm going to show you a picture. And the reverse. And the reverse is called as mesenchymal epithelial transition. Okay, so I would like to just explain one more thing is that if a cell goes through EMT, then it can also, this cancer cell has the property to go back and to go back into MET. So once it has passed into the blood, it can go and it can become, this mesenchymal cell will again go back and become epithelial, and that's a transition, and then it will go into a distant tumor. So So it is a vice versa process. So you can have EMT, and then after it has traveled, the cell becomes MET. So I hope you're understanding, but even if you're not, it'll come very quickly. It'll be easy. So the reverse can take place, and if you have both markers equal, if you have epithelial markers and you have mesenchymal markers, then that means it's a hybrid kind of a cell. So let's see what's next. So, so I just told you that cells can uh, take a mesenchymal transition and change their shape, and when they have traveled and they go to a distant site, after migration, they can again go back to becoming epithelial cells. They have it in them, so they become MET. Okay, so that's just the story here. Now, sometimes uh, this is like a big state. Either you're epithelial or you're mesenchymal. It's like, you know, it's like these cells, they have the capability to convert and to take on a different personality, a different temperament, and do the job, go somewhere, and then come back home and become again, not come back home, but go to a different location and then again become the original self all over again. So that's all it is. Epithelial and mesenchymal. It was original and then it becomes sneaky and it travels and then it finds another house and another location and then it becomes original all over again and settles down. So let's look at this slide now. For so so let us examine this EMT some more and and the cytology associated with it. So this is an example in this study. This is a mouse, okay? And this is a mouse, and what we're looking for in this mouse is oops, sorry. We are looking for EMT cells. We're looking for epithelial cells, and we're looking for mesenchymal cells. Okay, my and we're looking at the border of the tumor epithelium. Okay, at the border of the tumor and the stroma. So we create a tumor, and we are at the border of a tumor and stroma. What is the stroma once again? Stroma are blood vessels. It can be immunity cells, everything that we showed you yesterday. All right. And so everything in the environment. And so what we're looking for is if this is really true, if we have this transition, then I should be able to see that. And so this experiment was these experiment was performed in mouse, and in that mouse, these breast cancer cells, breast cancer epithelial cells, were implanted, and they were implanted into this into the mouse host, into an immunocompromised mouse host. And so then you just give it time, you grow it, you give it some days in this mouse, and so after a while, then you take a little biopsy, and from that biopsy, you do, like I said, you cannot do H&E stain, doesn't mean anything here. You have to do immunohistochemistry and or immunocytochemistry. And so you look for those cells. So you look for the kind of cells. And so they stained everything. And what they found is that the human, so these are the human cells. So this is the human cell. These are human cancer cells, right? And so what they found is that the red ones, okay, so the red ones which are staining for cytokeratin. The red ones are staining for cytokeratin. And what does that mean? And the green ones, let me just finish this up, and the green ones are for vimentin. Vimentin is mesenchymal marker, and cytokeratin is epithelial uh, marker. Okay, so how do we know that? Remember I said that epithelial markers are cytokeratin. Remember this up here. Epithelial markers are cytokeratin, and mesenchymal markers, mesenchymals are vimentin. So that's mesenchymal. So it's becoming mesenchymal. And so if it's mesenchymal, what should it look like? It should kind of look like this, but I don't know if that's possible to see all the time. So anyway, the green ones are mesenchymal. And so where do you see? So this is the full tumor. This is the full tumor. Look at this. This is the full tumor. Inside the the inside of the tumor is more epithelial. So here these are more epithelial. So this is epithelial. Red on red won't work. And on the sides, on the corners, near the stroma, look at this. All of these that are near the stroma, near the stroma. Okay, they are, look at this shape, it can even looks like that. The near the stroma, that has more vimentin, and the fact that it has more vimentin means that it is trying to break away and go into the stroma. So this one is a mesenchymal, and all of the ones that are in the center here, those are all epithelial. This is proof. All right, this is proof that the epithelial cells of this breast cancer human tumor are changing their shape, are changing their properties to epithelial because they have taken up, they are showing the dye called as vimentin. As they're showing the protein called as vimentin. So these are, oops, wrong labeling, sorry. These are, all the green ones are mesenchymal. So vimentin means mesenchymal, keratin, cytokeratin means epithelial. And they have lost, some of them have lost cuboidal shape, although not easy to see here. They have lost their epithelial shape, and they have assumed a more elongated fibroblastic shape, like a fibroblast. Alrighty, so this is one way to prove it. So not to confuse you too much, but I have another diagram here, and this is again immunohistochemistry, where now the reversibility, so if you're EMT, then after a while you can reverse. You can be reversed all over again. So how do you reverse? Okay, I'm going really, really slow today. So, how do you reverse? Well, then, in other places, people have done staining. Okay, they have done staining in a primary tumor. Okay, and they have done staining in a liver metastasis, and they have looked for, again, they have looked for cytokeratin. Cytokeratin is a epithelial state, and they have looked for loss of that. So they have looked for cytokeratin, and they have looked for basement membrane. Basement membrane is green. Basement membrane is also epithelial. So the hypothesis here is, the thing here is that if there is a primary colorectal cancer, so this is a patient actually, and a patient's tumor was taken and then it was stained by fluorescence dyes. So green, and so there, the hypothesis is that if the, in order for this primary tumor to go and to travel by EMT and to travel to another tissue, it should lose, it should not have any more keratin, and it should not have the basement membrane because it's breaking down the basement membrane. So here it is, your primary tumor. Look at this, number one. So I'm on number one here. Look at their basement membrane. There's so much of basement membrane here. Basement membrane, that is your basement membrane. These are green, all right, in this colorectal cancer patient, basement membrane. And there's a lot of cytokeratin. All of this red is all cytokeratin. Okay, so uh, over time, they probably are looking at a different tissue or nearby tissue, and it has undergone EMT, epithelial mesenchymal transition. The same tumor cell has changed, and the same tumor cells at the edge of that tumor, they stained it, and at the edge of the tumor, there is no more basement membrane. There's no green. Look at that. There's no green. The basement membrane has been broken down. Okay, a little bit of green, a little bit of green. The basement membrane has been broken down on the edge of that tumor, and that means that this and cytokeratin is not so much. The here on the number one, cytokeratin, a lot of red, but in number two, the red is decreased, cytokeratin is decreased, and this indirectly means that this is a mesenchymal transition. All right, so we have to use these markers right now. There's no other way to do it. So this is all mesenchymal transition. And guess what? They, in that same patient, they looked at the liver, where the cells have spread to the liver. So these are colorectal cells that have gone to the liver. And what they find is, again, they find the green. So that means this has undergone MET. So it has undergone MET transition. So from EMT, it goes back into the original. It will um, it will go back into the original, and it makes more basement membrane. It is synthesizing basement membrane. It is making or it allows the basement membrane to be synthesized if it's not making itself. And there's much more red. Look at this, much more red. Much more red means more cytokeratin. Okay, so this is a very direct way of showing. This is a very direct way of showing patient sample, and by doing immunohistochemistry, by showing, and fluorescence, showing that the cells have undergone EMT, the cells have undergone EMT, and they have degraded the basement membrane. They have degraded it as needed, and then it did it again. Okay, and it removed cytokeratin. So this is a reversible process. Okay, that means this is a reversible process, and it simply means that um, if we are able to catch these at an early stage, maybe we can start thinking about earlier treatment. So we said that the EMT process is so important. Again, the picture is here for you so that you keep on remembering. Look at this, the EMT. So just to remind you again, what was the EMT process here? The cells have shape, they have cuboidal, but when they go transition, they have lost polarity, they have lost their shapes. And honestly, we don't say that they have lost their shape so much. It's just that they have these variable shapes. So this is something that the cell did so far. So far you learned that the cells did it. But what if I told you that the macrophages can also do that? They can help. So this is the bad news. So breast cancer cell, okay, macrophages and okay, so there's a mistake here. Macrophages and breast cancer cells, they communicate and they tell each other. All right, breast cancer tells the macrophage to help me convert to EMT. So here's your breast cancer cell on the left hand side. It's a breast cancer cell, and it kind of like has these. Let's not talk about the this here. Let's not talk about the genetics here or anything. So this is your breast cancer cell. This is your breast cancer cell, and this is your macrophage. And the macrophage is supposed to try to destroy these cancer cells, but no, the macrophage here sends out the breast cancer cell. Sorry, let's just wipe this out. The breast cancer cell secretes a substance called as CSF. All right. And it secretes the substance, and the macrophage takes that information, and then the macrophage gives it epidermal growth factor, and so the epidermal growth factor binds here, and this breast cancer cell then even multiplies more. So it is a cooperation. They tell each other. The breast cancer tells the macrophage, breast cancer cell tells the macrophage to signals the macrophage to make more of substances such that it can keep growing, and it can keep growing, and it can turn into it. The diagram doesn't show you, but it can turn into EMT. So breast cancer cells and macrophages together. So the macrophage helps the tumor cells and makes it EMT, although I did not show it to you here. But what I am showing to you is on the left hand side, on the right hand side, in pancreatic tumors. Whoops. In pancreatic tumors. So these are pancreatic islet cell tumors in a mouse model. So now we're looking at a mouse model. It is not a patient here. And in this case, the macrophages are recruited. Macrophages are brought in. All right. And so the macrophage are being brought in, and they're being brought in at this dotted line. See that dotted line here? And the macrophages have been the macrophages are just shown here. They're shown here in the green, I think. Okay, the macrophages are shown here secreted because the macrophages make this protein. So this is a macrophage, this is a macrophage, and all the macrophages are traveling in this direction. These green macrophages are traveling in this direction, so we label them green. And what are they doing? So these are your tumor cells. Look at these tumor cells. These tumor cells where they're close to the macrophages on this side, on the second bottom here, on the this bottom location here, you see that there is none of the ecadherin. Ecadherin is in red. These are ecadherin. What is ecadherin? It helps to hold cells together. These are all ecadherin. And so here these cancer cells are held together, but look at here in this area, there is no ecadherin. They're all blue. Okay, there is no, this red is just not there. It's everything is blue. Everything is blue. Why is it blue? There's no ecadherin. And how is this happening? So the understanding is this is also happening not just by the cancer cells, but also by these macrophages that are also coming up, and they are coming up and they are secreting things such that it becomes EMT takes place. So how do you know that EMT took place? Because ecadherin is being lost. Ecadherin is being broken away. And cells are breaking loose. And look at this. Cells are also kind of like breaking loose. This is a loose cell. This is a loose cell. So now we move to the next parts, and we can just take these a little more quickly. And so in this metastatic process, next is to go into the blood vessel. So it's going to go into the blood vessel. The tumor cells now have changed their shape. and they can enter the blood vessels, and so the basement membrane was all degraded by these MMPs, etc. And those these cells will travel, and they might make new blood vessels later at some point, or they may not make new blood vessels. So the bottom line is, now there is intravasation. It enters, all right, enters, and if it's possible that it can also help create. Okay, it's giving off so many signals here as it's going that it might make some more branches. So it might make more branches. All right, so let's go to the next. So then it goes transport, and it can transport in through the blood. And these cancer cells, they cannot keep on traveling and traveling in the blood. It's just not a thing. They need to they need to hold on to something at some point. So they need some kind of anchorage. And if they don't anchor at some point, they will just die. So they will just die. And so the blood is not a very good environment. So just flowing in the blood is not a good thing. The blood is a actively hostile environment for these cancer cells. So take a look at this diagram. It is, while it is trying to survive and secrete things, for the most part, the blood vessel is not a very good place. And it needs to not travel through the blood for too long. It needs to come out, and it needs to come out at some point very quickly. All right, so if those the tumor cells, which are fairly they're 30 micron size, and if it enters into a small capillary which has a diameter of 8 microns, then that means some of the tumor cells can get trapped, okay, in the capillary beds that they encounter first. So they encountered the first, okay, the first pass organ. And that's why the organ has to be nearby. And so this is how they will now, if they get stuck. So now this is just to show you where all the circulation is. I'm not going to go and walk you through the whole circulation process. But this is where the cells now need to either make a tumor there, or they need to come out of the blood vessel, sneak out of the blood vessel. Okay, so they will now, the steps are same like intravasation, but things are just now going to be like opposite, they're going to sneak out. So let's just see how that happens. So here are your circulating tumor cells, CTCs, all right, and they're undergoing extravasation. And so what is that whole process of, just in a simple diagram? So here it is, it's stuck on the left hand side. It is stuck in a narrow capillary. It is stuck in this capillary, and that's a good thing. So now the platelets will come, all the platelets will come because they see a foreigner. The platelets, that's what they do. They see something foreign like this, they will start to stick. And then it will attach, if it can attach fairly quickly. And then look at this, it's going to, make some blood vessels of choice here. So it's going to make some blood vessels if possible. All right, and attach to the capillary basement membrane. And then at some point, number 4, it is attached to the basement membrane. At some point, the platelets, it will remove the platelets. It will allow for the platelets to be like comfortable, and they leave. They are not in touch anymore. And then this is where it just multiplies, and then it extravasates out from the blood vessel. And so I have this imagery for you. It's another type of various kinds of imaging, and then I think I have a little video for you. So essentially, these tumor cells, they are now passing through the circulation, and this is just a beautiful, what kind of a intravital microscopy here, showing you this is the E is here. This is the blood vessel. This is the blood vessel. And R is a red blood cell. This is a red blood cell. Okay, so there's a line here. That's like your blood vessel, if you can see that line. and this is your white blood cells, okay? And they are, let's see, leukocytes. So just to show you that the diameter, this is the diameter. In this diameter, cells can get stuck. It's just to show you that, okay? So cells can get stuck in here. And so this is a special kind of a, here, this is a special kind of a diagram to show you that this everything has been dyed here. And so it's just to show you that where is it? That the red blood cells are deformed. The red blood cells here, they are passing through the blood vessels. See those red blood cells? They're passing through the blood vessels. So that's your red blood cell here. These are your red blood cells, fairly deformed, and that some cells that are not deformable, they cannot pass through these passages, and then blood vessels have to be made. Okay, but this is the diagram that you want to see now, that because of this narrow passage, these are the green cells. They get stuck at certain points, and they have formed a they have attracted other cells. They have multiplied a bit. Whoops. And they have attracted some some platelets, and they are now found attached. So they are found in the blood vessel, and then they are stuck in the blood vessel. And so this is a prostate cancer patient. Okay, this is a prostate cancer patient in which they have been able to recover this whole thing from the blood vessel. And then what else? The rest is formation of micrometastases and then colonization. So I do have a nice video to show you that answers all of this. Okay. So the bottom line and the full story is that metastases doesn't occur so much. Okay. There is a low rate of success. Okay. There are cells that are getting rid of it. So there's a low rate of success of forming metastases, but it happens. So therefore, it can take a long time. And the earlier steps of the cascade, going into the blood vessel, that can be efficient, but the last step of colonization, it's just not so comfortable. It just doesn't happen so easily. All right, so the complete scenario is here. This is the complete picture. There is carcinoma in situ, and progression. And then some of them, they change into EMT, they change their shape, etc. They invade, and then from there they go, and the stroma is also helping them to do all of that, reactive stroma. Then they go into the blood vessel, so they're migrating through the blood vessel. This is the mesenchymal property. They transport into the blood vessel, and look at this. They are exit the blood vessel because they can sneak out, and that's extravasation. And when they are able to sneak out, and then they are close to next to the tissue, then they go MET. So from EMT, they go back into their form, and then they become more cuboidal, and then they attach to this basement membrane where possible, and they probably take the help of the stroma. But here, this is a normal stroma. The stroma doesn't have to do anything. This stroma doesn't have to do anything. Here it was a reactive stroma. Stroma was helping. Here, nothing to do. It will just simply go and attach, but it takes a long, long time for this colonization to take place because the stroma is not doing anything, and the likelihood for it to survive is also important. So colonization takes it's with some difficulty. So with this is the whole story. EMT, you as a computer science student should be able to see how there are ways to look for EMT without all that staining. And so are there data sets? Maybe you can find that they're clearly much more visible and detectable early on. Okay. So I'm going to take you to two videos. One video is that the brain metastasis requires 4 steps. It's kind of simple, and that's needed for colonization. And the second one is that if there are cancer cells, there are cancer cells and they have been put in the brain of a patient, form it so easily. It's a process, but it's not so easy. Okay, so those are the two things. Now I have not asked any question yet. Are you all with me so far? And
Yes, ma'am.
Or the
Yes, ma'am.
formation of malignant growth at a site of the body far removed from the location of the primary tumor, is responsible for 90% of deaths from cancer. In this image, we see multiple breast cancer metastases to the brain. Multiphoton laser scanning microscopy, together with image processing software, can be used to image metastases to the brain of a live mouse. A transparent window permits visualization of the various steps of the brain metastatic cascade that we will observe in this movie. The brain metastatic cascade can be broken down into 4 steps. 1. Initial arrest of circulating tumor cells in narrow microvessel.
Maybe there is a problem.
2. Escape of
Video is
arrested
not
cells from the microvessel into the brain parenchyma, process of extravasation.
video is not there, ma'am.
3. The establishment of the recently extravasated cancer cell in a location close to a blood vessel, referred to as the perivascular position. 4. And finally, 2 alternative routes of metastasis formation, either cooptive growth, in which cancer cells grow along
Oh, ma'am,
existing blood
excuse me,
vessels,
ma'am.
or
yeah, hold on.
angiogenic growth, in which cancer
Ma'am,
cells actively
if
induce
there
the
is any
formation of new.
video playing, the video is not playing properly, ma'am.
Oh,
It is only on one screen is showing.
the videos. So what are you looking at right now? What do you see?
A yellow color
Oh,
something, a cell type.
okay. Let me see if maybe I'm doing it the wrong way. Now, do you see?
Yes, ma'am.
Thank you for alerting me.
Metastasis, or the formation of malignant growth at a site of the body far removed from the location of the primary tumor, is responsible for 90% of deaths from cancer. In this image, we see multiple breast cancer metastases to the brain. Multiphoton laser scanning microscopy, together with image processing software, can be used to image metastases to the brain of a live mouse. A transparent window permits visualization of the various steps of the brain metastatic cascade that we will observe in this movie. The brain metastatic cascade can be broken down into 4 steps. 1. Initial arrest of circulating tumor cells in narrow microvessels. 2. Escape of arrested cells from the microvessel into the brain parenchyma, the process of extravasation. 3. The establishment of the recently extravasated cancer cell in a location close to a blood vessel, referred to as the perivascular position. 4. And finally, 2 alternative routes of metastasis formation, either cooptive growth, in which cancer cells grow along existing blood vessels, or angiogenic growth, in which cancer cells actively induce the formation of new blood vessels to nourish them. In the examples that follow, melanoma cells took on cooptive growth, whereas lung cancer cells were found to take on angiogenic growth. Here we see the first step involving the initial arrest of a cancer cell, labeled red, and a capillary, labeled green. A second cell is later seen above, trapped in an even smaller vessel. The second step in the brain metastatic cascade is the extravasation of cancer cells from the lumen of the capillary. Cancer cells use several alternative strategies to extravasate. In this movie, we see a cluster of cancer cells extravasating, one after the other, out of vessel number 1 into the brain parenchyma. The third step involves the establishment of perivascular positions, which seem to ensure the initial survival of the extravasated cells. This movie shows a tomographic image of cancer cells in perivascular positions. Nonetheless, assumption of a perivascular position does not on its own guarantee long-term survival, as seen in these images. Although this micrometastasis grows in a perivascular position for the first 9 days, it is completely gone by day 14. The fourth and final step in the brain metastatic cascade involves two alternative strategies: cooptive growth or angiogenic growth, depending on the type of cancer cell that has metastasized. As mentioned previously, lung cancer cells initiate angiogenic growth in order to nourish themselves. In this series of images, we see how the potent angiogenic powers of lung carcinoma cells are able to provoke the florid outgrowth of microvessels that sustain the active growth of the tumor. An alternative strategy for growth is exhibited by these melanoma cells, which have disseminated to the brain and assumed a perivascular position. Here, we see how their expansion has depended on the cooptive strategy, involving growth along the outside of blood vessels. The multiphoton microscopy used to create these movies demonstrates the extraordinary advances in the powers of modern imaging technology to study cancer in living tissue.
Yes, ma'am.
Yes, ma'am.
Okay. Alrighty. So just
Metastasis has generally been a difficult process to visualize. In this model, high-resolution magnetic resonance imaging, or MRI, has been combined with tomography techniques to visualize the metastasis of breast cancer cells to the brain of a mouse. Before the injection of cancer cells into the circulation of the mouse, the cancer cells were labeled with a dilutable label, that is, one that loses half of its intensity each time a cell divides. This dilutable label is seen here in red. Following initial injection at day 1, many hundreds of labeled cells are apparent in the brain. In the succeeding days, the number of these cells that retain full label, and therefore have not divided, is diminished, indicating the gradual elimination of these cells over the subsequent 3 weeks.
So essentially, they've just been removed. Those cancer cells, they have been killed or destroyed.
At the beginning of the 3rd week, small metastatic growths suddenly begin to appear in green. The green signal represents actively growing cancer cells. These green metastases continue to grow in size, while the individual non-growing single-cell metastases continue to disappear. Among other lessons, this experiment illustrates that hundreds, if not thousands, of micrometastatic single cells may be seeded initially in a target organ by cells arriving from the primary tumor via the circulation. However, only a minute fraction of the initially seeded cells succeed in spawning metastatic colonies.
So the beauty of this is that it's not so, it's a hard life for a cancer cell. I guess that's what it means. So, not to make it funny, but where did it go? Oh, was that it? How did I end up here? So those were the two videos, and I guess we will just, I will just end it with a little bit of a summary. So colonization is not so easy. Vascularization is also not so easy. And so this is a diagram that just shows you that not every primary tumor has a tendency to flourish. You know, there is something called as metastatic tropism. And all it simply means is that there's a preference from what goes where and which one can go to a distant site. So the relative width, the thickness of each arrow here indicates a proportion of how much metastases can occur. So if it's prostate, then a very much tendency for bone marrow. If it's prostate, even less tendency for brain and lungs. However, we just saw, for example, if there's breast cancer at the bottom here, then it can go towards the lungs more, but breast can give rise to bone marrow bone marrow cancer. So uh, this is a nice diagram to say which are the more tendency and which is the less tendency for metastasis to take place. So for prostate, breast, pancreas, and colon. So people have observed this and tracked this, and therefore we have this information. So I think kind of like everything, like at this point, these questions are for you. So in terms of cytology, what is the challenge for EMT? Um, well, for EMT, it's a heterogeneous mix. Those cells, they are um, they are not completely E and they're not completely M. They're not completely epithelial and they're not 100% mesenchymal. So they exist along a spectrum. So, they're probably transitioning. And so that is very dynamic. And so if it's this dynamic, then there is difficulty to capture that in a static sample. All right, so that's why maybe in one picture, in a static sample, it's just hard to find if they're existing in a spectrum. If it was a switch on and off, then that would have been one thing. You can have only E and only M type. So therefore, just having morphology alone is not good, is not enough. So just by having pure morphology, appearance based analysis is not correct. This does not work. Okay, that does not work. And so you have to do molecular or immunocytochemical methods. And therefore, today we had to talk about proteins because if you're going to do immunocytology, then you have to target a protein. And you know that now that cytokeratin, vimentin, that they have some meaning. Cytokeratin is for epithelial, vimentin is for mesenchymal. And so then there's intermediate states. There's like both of them. Some of the cells, they don't look like they have a different shape. They are a hybrid. Okay. So they're in between. They're like E/M phenotype together. And why? Because they co-express both markers. And that's more complicated now. So if they're going to co-express vimentin and cytokeratin, not either or, because it's 50-50, then that's much more complicated. So you see how complicated it is to do this cytology. So I end this talk with all the techniques that are used for EMT. Why EMT? That's the major, really important process that governs how smart metastasis will be, if metastasis will take place. So these are the techniques, and I'll just, I'll just walk you through this one page. So this is, we just know that these are two, they're already there, and I just added those names of those proteins for you. Flow cytometry is another one, and it has limited application for patient tissues. So one can do flow cytometry, but we really do it for basic research. And you can use a laser beam to, to count the number of cells and to examine the cells. But it could be done for research. Multiplex imaging is a much more advanced method, but I'm not sure how that method kind of works. But it can be used for both EMT and MET states. Forgot what the method was. Oh yeah, multiple fluorescence markers. So yeah, I showed you already two fluorescence markers I already showed you. So that can be done. So, but this is, yeah, it's combined with mass cytometry. So in lung cancer, they have used mass cytometry to identify these different states in patient-derived samples. And the best one was really intravital live microscopy, but I think that this is also, at this point, it's only used for preclinical animal models. Okay, so it's only to visualize the transitions. Okay, so if there are data sets for you, typically that should be here. They could be data sets for you. What are your challenge? What are your you could look for leader cells. You could look for early EMT cells or early MET cells, and you can try to see if you can identify, or the machine can identify any of these fine differences. and you need to identify what features you're going to extract for that also. So with that, I end this talk, and I will take any questions that you have. So Pooja, take it away. I'm done.